In a quiet pond, there is a single-celled organism named Tetrahymena that seems to have forgotten how to die. It just divides, endlessly. This simple fact presented a massive puzzle to biologists like Elizabeth Blackburn in the 1970s. Because every time one of our cells divides, its DNA, the very blueprint of its existence, gets a tiny bit shorter at the ends. Imagine photocopying a page of a book, but the machine always cuts off the last word of the last sentence. After 50 copies, you'd be missing a whole paragraph. Our cells face this exact same end replication problem. So how did this microscopic creature in a puddle of water manage to cheat the system? The ends of our chromosomes are capped with long, repetitive, non-coding sequences of DNA called telomeres. The easiest way to think of them is like the plastic tips on your shoelaces, the aglets. As long as that aglet is intact, the shoelace is protected from unraveling. With each cell division, a small piece of that protective aglet is lost. The important genetic code, the shoelace itself, remains undamaged for a while. But after 40 or 50 divisions, the telomere is worn down to a critical point. The cell senses this impending damage to its essential DNA and does something remarkable. It stops. It enters a state of permanent retirement called senescence. The solution discovered inside Tetrahymena was an enzyme that meticulously rebuilds these aglets. They called it telomerase. It is not just a simple protein, it's a sophisticated molecular machine. It's a reverse transcriptase, meaning it builds DNA from an RNA template it carries with it. This is backward. The central dogma of biology is that information flows from DNA to RNA. In fact, this reverse strategy is most famously employed by retroviruses like HIV to stitch their own genetic code into their host's DNA. The tool for potential cellular immortality operates using a tactic that is more commonly associated with a viral invasion. This raises an obvious question. If our bodies have the gene for this incredible repair enzyme, why is it switched off in most of our cells? Why do we have this built-in aging clock? It turns out that this limitation is not a bug, but perhaps the most important feature of our biology. An essential part of the definition of cancer is uncontrollable cell division. The shortening of telomeres acts as a critical tripwire. It ensures that a normal cell can only divide a set number of times before it is forced to shut down. In a very real sense, your cells age and die to prevent you from developing cancer. We are designed to grow old. However, some of your cells do use telomerase. Your germline cells, the sperm and eggs that create the next generation, must be perfect. Their telomeres are constantly maintained. Certain populations of adult stem cells, the body's internal repair crew, also keep telomerase active to allow for tissue regeneration throughout your life. But there is a third, more sinister type of cell that reactivates this enzyme. About 90% of all human cancers manage to find the switch to turn telomerase back on. This grants them a form of biological immortality, allowing them to bypass the aging tripwire and divide forever, consuming the body from within. If telomerase is so critical, do animals with longer lifespans just have more of it? The real world is rarely so simple. Take the naked mole rat. This strange, subterranean rodent can live for over 30 years, an eternity for an animal its size, and is remarkably cancer-resistant. Its cells maintain high telomerase activity throughout its life. But then you have the blue whale, a 90-year-old behemoth whose telomeres appear to shorten just like ours. Even stranger, the common house mouse, which lives only 2 or 3 years, is born with telomeres that are 5 times longer than a human baby's. Clearly, there isn't a simple equation where more telomerase equals a longer life. So, what if we developed a drug that could safely turn on telomerase in every one of our cells? Would we become biologically immortal? Perhaps. Our tissues might repair themselves perfectly. Our organs could remain youthful. But we would be accepting an immense risk. Every single one of our trillions of cells would now have a key defense against cancer permanently disabled. The mathematical probability of a random mutation leading to a tumor would skyrocket. We would trade the slow, predictable process of aging for a life of playing Russian roulette with cancer. Our limited lifespan might not be a problem to be solved, but an elegant, hard-won evolutionary compromise. Ultimately, telomere shortening is just one piece of the vast, complex mosaic of aging. Your body wears out for 1,000 different reasons, all at once. There is the constant accumulation of DNA damage from background radiation and the simple act of metabolizing food. There is the slow, inevitable decay of our mitochondria, the power plants in every cell. Epigenetic changes cause our cellular machinery to read its own instructions incorrectly. Activating telomerase would be like patching a single crack in a dam that is crumbling from top to bottom. The protein that protects the ends cannot stop the entire structure from getting old.
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.